Hello and welcome to our lecture today on peptide hormones, synthesis, secretion, and signal transduction pathway. This is a very important lecture, and our objective is to identify the processes that lead to the synthesis of a peptide hormone and the definition of what structurally constitutes a peptide hormone and what are examples of peptide hormones. And once these hormones were synthesized, stored, and then secreted, once they are secreted in the blood, how are they going to be transported? And once they arrive to the target destination, the target cell, and they bind an extracellular receptor, what happens underneath? What is the signal transduction pathway that manifests in the cytoplasm on the other side of the interface of the cell membrane that culminates in a target response in the target cell? Very, very, very important lecture. So we must first begin by defining a peptide hormone. Obviously, there are only two types of hormones. If you are a hormone, you are one of two. You're either a steroid hormone made up of lipids, or you are a peptide hormone. If you are a tiny peptide hormone, meaning you are below 100 amino acids, that makes you a polypeptide hormone. But if you are in number more than 100 amino acids, if the polypeptides are more than 100 amino acids, that is no longer a polypeptide, that is a protein hormone. So the difference between a protein hormone and a peptide hormone is in the number of the polypeptide. Both of them, the protein hormone and the peptide hormone, are both made up of polypeptides, connected connected amino acids. But the number is what makes the criteria and the classification. If the amino acids happen to be from 1 to 100 amino acids, that particular hormone happens to be a peptide hormone. If the amino acids happen to be above 100 amino acids, as in 101 amino acids, that makes it a protein hormone. But in the end, a protein is a protein, a peptide is a peptide, but they're all made up of amino acids with a carboxylic terminal and an amino terminal and an H group and an R group. But in the end, amino acids that are polypeptides or proteins, they are lipophobic, they are hydrophilic. That makes perfect sense because we have an amino terminal and a carboxylic terminal, and that enables for the dissociation and the dissolving in the polar substance. So that's why peptide hormones do not need a carrier in the blood. They can because they can dissolve in the blood. The blood is polar. The blood is plasma. Blood is made up of red blood cells and the buffy coat, which is white blood cells and platelets, and then you have the plasma. Plasma is full of electrolytes, proteins, and water. That's the perfect medium for a non-steroid hormone, for a peptide, which is a peptide hormone. A peptide hormone, non-steroid hormone, amino acid hormone, they have no problem dissolving. The problem is with the steroid hormone. The steroid hormone does not have an amino terminal. The steroid hormone does not have a carboxylic terminal. The steroid hormone is made up of lipids. So lipids do not dissolve in the blood because the blood is polar. Lipids are non-polar. Non-polar will never dissolve in polar blood. So how do we solve the problem? We solve it with a lipoprotein, a carrier. So the lipoprotein from the outside has phospholipids, polar heads, and in the core, the micelle has a hydrophobic nonpolar core. So the nonpolar core will harbor in its core this non-steroid hormone, this steroid hormone, to carry to the target destination, and then that steroid hormone would dissolve in the lipid membrane and cross the bilayer leaflet so smoothly, and then it will swim in the cytoplasm and then bind the receptor, bind the steroid receptor either on the cytoplasm, and then mobilize to cross the nuclear envelope and then enter into the nucleus and then bind the promoter region of the DNA to transcribe for us mRNA, and that mRNA will be translated into proteins. Now, the receptor could either be in the cytoplasm, the steroid receptor can either be in the cytoplasm or it can be in the nucleoplasm. But in the end, they call it the mobile receptor hypothesis, where the steroid receptor would bind into a receptor that is either in the cytoplasm or in the nucleoplasm and then migrate and move to the promoter region and transcribe for us our mRNA to give us our target protein. Okay, now that was steroid hormone. The other criteria, the other structural classification, so we classify hormones based on structure. We have hormones that are steroid hormones. Their structure is steroid, meaning built based on cholesterol. The cholesterol has 4 rings. It's hydrophobic and it's lipophilic. So they can, like we said, we just talked about the mobile receptor theory. However, the peptide hormones, they cannot enter because the bilayer leaflet is made up of nonpolar core. So this nonpolar core will prevent the catecholamines from entering. So we solve this problem with, if we say this is the cytoplasm, this is the bilayer leaflet, and this is the extracellular environment, there is a receptor with a head that is on the outside, and then you will have your catecholamine and your peptide hormones bind on the extracellular region, and then you would have a signal transduction pathway that requires a second messenger to manifest for us the target response. But with the steroid hormones, you will never need a second messenger, because the first messenger can cross the bilayer leaflet and it can also cross the nuclear envelope. Okay, so today we will talk about peptide hormones. Peptide hormones, so all hormones, whether peptide or not peptide, how are they synthesized? They are synthesized based on transcription. So obviously amino acids are the building blocks of proteins, and you will never get amino acids except from the ribosome, because the ribosome will synthesize amino acids for us based on an mRNA prescription, based on an mRNA transcription. And that mRNA transcription, we can never get it except we do transcription in the DNA. So this is called the central dogma of biology, the central dogma of biology. What does the central dogma of biology say? The central dogma of biology says that we translate mRNA to get proteins. And before that, we transcribe DNA. So we have transcription. We transcribe DNA to mRNA. Then we have translation. We translate mRNA into proteins. We translate mRNA into proteins. So if these non-steroid hormones, these peptide hormones, happen to be made up of amino acids, then we can never get amino acids, which are building blocks for proteins, without transcription. So we will first transcribe in the DNA, then we would send a nascent mRNA transcript. This is the endoplasmic reticulum, here in purple, the endoplasmic reticulum. The endoplasmic reticulum has embedded ribosomes. These ribosomes are very important because they will translate for us the nascent mRNA transcript. And they will create for us a pre-pro hormone. A pre-pro hormone. What's a pre-pro hormone? A pre-pro hormone is a draft of the final hormone. It's not yet ready. It's not yet ready. So we go into the ribosome, and the ribosome the ribosome in the ER, endoplasmic reticulum ribosomes, and that endoplasmic reticulum ribosome will cleave the pre part and leave us with a prohormone. Then we would move on to this brown region, that's the Golgi body. And there in the Golgi body, we will undergo even more modifications, and now we will liberate the pro part, and we will have our final hormones stored in vesicles. And then once we have the proper stimuli, the vesicle is going to open up and extrude its content, that hormone, outside in the extracellular environment. So that's very important for you to understand how peptide hormones are made. And remember, the emphasis is on endoplasmic reticulum ribosomes. There are a lot of ribosomes swimming in the cytoplasm. Why can't we utilize the ribosomes that are free-floating ribosomes in the cytoplasm? Because these ribosomes are to make proteins that will be used locally inside the cell. But proteins that will be extruded outside of the cell, they have to go to the endoplasmic reticulum. That's a very important distinction for you to find out the difference between endoplasmic reticulum ribosomes and ribosomes that are freely floating in the cytoplasm. So these endoplasmic reticulum ribosomes will synthesize for us a pre-pro hormone, a hormone that is a draft, not yet ready. It needs to undergo modifications and activation. Then after the endoplasmic reticulum gets for us a prohormone, then the Golgi body will take the prohormone, cleave it, and make it a ready hormone stored in these vesicles, and upon the proper signal, it will be sent to the extracellular fluid outside. So now this is happening for all of our peptide hormones. Every single peptide hormone is here. Now the problem is when this peptide hormone gets to the target cell. So now the peptide hormone has been extruded into the extracellular environment. It is now extruded into the extracellular environment. Once it binds to the target cell, once our hormone binds its target cell. Once our hormone binds its target cell, what are the effects? What are the effects of our hormone? So now we're no longer talking about a hormone being synthesized, in the endoplasmic reticulum, ribosomes, and then stored in vesicles of the Golgi body, and then extruded into the extracellular environment. Now we're talking about this hormone swimming in blood and reaching the target cell. This is the target cell. So obviously it's no longer a secret that these catecholamine peptide hormones are charged, and because they are charged, they can never cross the bilayer leaflet. And since they can never cross the bilayer leaflet, they can only bind on the outside on an extracellular receptor. And that extracellular receptor, once it binds to the hormone, to that ligand, an intracellular signal transduction pathway will happen that will manifest for us the target response and the target effects of that particular peptide hormone. So now let's look at the peptide hormones. What do they do? Well, if a peptide hormone binds on the outside, what happens inside is different. So we will have one messenger. So in the steroid hormone, we have one messenger doing everything. One messenger, we just need to carry it in blood because it cannot dissolve in blood. We need the lipoprotein to carry it. But once it got there, the first messenger will cross the cell membrane, will swim through the cytoplasm, will cross the nuclear envelope, swim through the nucleoplasm, bind to promoter region of the DNA, transcribe for us our target protein, mRNA, and then the mRNA will be translated into the endoplasmic reticulum ribosomes, and then the endoplasmic reticulum ribosomes will secrete for us vesicles full of prohormones, and these prohormones will undergo some post-translational modifications in the Golgi body, and then the Golgi body will extrude the content of this vesicle into the extracellular fluid. Now, the catecholamine can never cross because it has a polar charged amino acids. So it will bind on the outside. What happens on the inside is one of three things that can happen. So once our catecholamine or peptide hormone binds on the outside, the fate will be one of three things. We can either have, these are the different second messengers. We can either have a calcium calmodulin second messenger. That's the first chance. Second chance, we can either have an enzyme linked receptor, or number three, we can have a G protein coupled receptor, GPCR. It's either one of those three. So now let's talk about each one in particular. Let's begin with the calcium calmodulin. With calcium calmodulin, you have to understand that calcium is exuberant, and there is a lot of calcium on the extracellular environment. There is a lot of calcium on the extracellular environment. There's a lot of calcium on the extracellular environment. So what are the forces that tempt calcium to move from the extracellular environment to the intercellular environment? Well, we have two forces. We have an electrochemical. That's force number one, electric. Force number two, chemical. So now, let's look at, we know that there's a lot of calcium on the outside. Let's compare outside to inside of the cell. Outside of the cell, outside of the cell to inside of the cell. On the outside, there is a lot of calcium. That means there is a lot of positive charge because calcium is positively charged. On the inside, there is a little bit of positive charge. So there is more positive charge on the outside and less positive charge on the inside. So now there's an electric potential for the movement of calcium from outside to inside. That's the first gradient to be fulfilled. There is a second gradient to be fulfilled, and that is the chemical gradient, where you have calcium atoms, actual atoms, calcium, calcium, calcium. There is a lot of calcium molecules outside. There is a little bit of calcium inside. So there is chemical gradient. That was charge related electrochemical gradient. This is a diffusion gradient, where a lot of molecules will have a lot of kinetic energy to move from an area of a higher calcium concentration to an area of a lower calcium concentration. So the gradient is there to move from the outside to the inside. We just proved that there is an electrochemical gradient. But just because the gradient is there, does that mean calcium will be so easily able to flow inwardly? That's impossible, because the calcium channels are closed, as you see here. This is green here represents calcium. So calcium cannot enter. Calcium cannot enter into the inside of the cell. Calcium cannot enter, so it will deflect because there is no permeability for calcium. Once the peptide hormone binds to the extracellular region of the receptor, that will lead to the opening of the calcium channels, and calcium will start flowing down electrochemical gradient to the inside of the cell. Once calcium is in the inside of the cell, there is a very important receptor known as calmodulin. This calmodulin receptor is inactive. This calmodulin receptor is off. is inactive. But when calcium comes in, calcium is going to bind calmodulin. So now you will have calcium, calmodulin. You will have calcium calmodulin complex. So you will have calcium calmodulin complex. This calcium calmodulin complex will now go and will activate for us target enzymes, turn them from off into on, from inactive into active, from not working into working. And that activation of these target enzymes will manifest for us target clinical effects. So here we need a second messenger, the calcium calmodulin second messenger, to mediate for us what this peptide hormone that is unable to cross. We did its effect not by a first messenger, by a second messenger, here being the calcium calmodulin. Once the reaction is over, calcium will separate from calmodulin, and then the channels now, and then when the receptor leaves, the channels will close, and then there will be no more calcium to bind calmodulin, and that will lead to calcium calmodulin complex to be off. So now we're done with the first one, the calcium calmodulin complex. Now let us move on into the second messenger, which is the enzyme link receptor. There are receptors. When the hormone binds now to the extracellular region of the receptor, what happens is that the channels will not open. That was just for the case of calcium calmodulin. Here what happens is there are enzymes that are linked to this receptor, such as the JAK enzyme, Janus kinase enzyme. This Janus kinase enzyme, anytime you see a kinase, that's a special type of enzyme that phosphorylates. So if you have a substrate and you add to it a mysterious enzyme, and then you examine the substrate and you see that this substrate now changed. It is the same substrate, but it now has an extra phosphate group, an extra PI group. You know what this mysterious enzyme is? It's a kinase. So a kinase enzyme is an enzyme that adds a phosphate group to its substrate. So whenever you know that this Janus kinase is activated, it will have one of two things. Either it will phosphorylate. Either way, it will phosphorylate. Phosphorylate. It will add a phosphate group. Phosphorylate. Case 1, it will phosphorylate. Case 2, it will phosphorylate. That's what it does. It phosphorylates. But what does it phosphorylate? Case 1, it will either phosphorylate for you target enzymes in the cytoplasm, and these target enzymes will give us target effects, target responses. Or it will phosphorylate for us a special type of protein known as STAT. STAT means S stands for signal, T stands for transducer, A stands for activator, and T stands for transcription. So you know transcription happens only in the nucleus. So you know that phosphorylation of STAT protein, which is signal, transducer, and activator of transcription, this phosphorylated STAT will migrate into the nucleus, and it will bind a promoter region, transcribe for us target mRNA. This target mRNA will go into the endoplasmic reticulum, ribosomes. It will be translated there to give us our pre-pro hormone. Then the pre-pro hormone is going to go into the Golgi body to undergo some additional post-translational modifications, and then we will have our pro hormone ready to be secreted. So these are the two effects of the Janus of the enzyme linked receptor. So now we're done with here we see that the binding of the hormone to the receptor led to the activation of a linked receptor, and the linked receptor and the linked enzyme to the receptor gave us our target effect. The last second messenger is the G protein system. So now we were done with two. Now let's go to the G protein. The G protein is itself divided into two parts. The G protein receptor, the G protein receptor, the G protein coupled receptor is divided into two parts. The first one is the G stimulatory and G inhibitory system, and the second part is known as phospholipase. Lipase is a enzyme that hydrolyzes lipids. So whenever you see lipase, that is an enzyme that hydrolyzes the breakdown of lipids. So phospholipase C, denoted with C. So now let's talk about the first type, G protein receptor, GS and GI. Why do we say GS? GS stands for G stimulatory. G stimulatory. G stimulatory. G stimulatory system, while GI represents G inhibitory system. G inhibitory system. The G stimulatory system and the G inhibitory system. Let us now talk about them in more detail. The G stimulatory and the G inhibitory system, their effects are different in the end, but the signal transduction pathway is very similar. So what happens with G protein coupled receptors, obviously why they call them G protein coupled receptor is because the G protein is attached to the receptor. So this is the G protein, this is the G protein, and in this case it's a GS, G stimulatory protein. The G stimulatory protein is bound to an alpha subunit, a beta subunit, and a gamma subunit. And it's also bound to GGP. GGP. What does GGP stand for? It stands for the nucleotide guanine dinucleotide, meaning two phosphate groups, dinucleotides. Guanosine dinucleotide. When the hormone binds the receptor, guanosine dinucleotide will be hydrolyzed into guanosine trinucleotide, GTP, guanosine trinucleotide. Guanosine trinucleotide will be attached to alpha subunit, and the GTP alpha activated the GTP alpha complex. So now we'll have a GTP alpha complex. This GTP alpha complex is going to go and activate for us adenyl cyclase. Adenyl cyclase was actually off. It was not working. It was inactive. But GTP alpha is going to hydrolyze adenyl cyclase from off into on, from not working into working, from inactive into active. And then once adenyl cyclase is activated, it will cyclicize for us ATP, which is adenine triphosphate, adenine with 3 phosphates, adenine triphosphate. It will turn it into AMP, adenine monophosphate. This adenine with 1 phosphate, after we subtracted 2 phosphates from it, this adenine monophosphate is going to go and activate for us a protein kinase, and obviously a protein kinase will be turned from off to on, and that means it will phosphorylate for us regulatory target enzymes, and these regulatory target enzymes will give us our target effect. So that will lead to stimulation, plus that will lead to activation of target response and upregulation of target gene transcription. In a GI, the same thing will happen, but in the end, so we will have GDP hydrolyzed to GTP, and GTP alpha will go and activate adenyl cyclase. Adenyl cyclase will signalize ATP into AMP, and AMP will activate for us protein kinase A. Protein kinase A will phosphorylate for us target proteins, but the target proteins, when compared to GS, will now, instead of activate target effects, they will depress, they will shut down, they will inhibit target effects. So in the cell, we don't need hyper mechanisms, we don't need hypo mechanisms, we need a moderation. So when the cell wants to press the brake, it will activate GI, but when it wants to perform an acceleration, it will activate GS. So, and then in the end, after we reach our target response, GTP will separate from alpha and will be hydrolyzed back to GDP, and then GDP will go back to bind the GS or the GI protein, and once GDP is back, alpha goes back to its old friends beta and gamma, and now we will be in the off position. So that's as far as the GS and the GI system. Now let us now look at the other G system, that is the phospholipase. The phospholipase, we said that it's an enzyme on the cell membrane that is usually in the off position. But once the receptor binds the ligand, it will be turned from off to on, from not working into working, from inactive into active. And once phospholipase C is active, it will grab a chunk, a phospholipid in the cell membrane known as PIP2. PIP2. What does PIP2 stand for? Phosphatidyl inositol 4,5 biphosphate. It will take PIP2 and break it into two target second messengers. It will break it into it will break it into IP3, we'll break it into IP3, and we'll break it into DAG. What does IP3 represent? IP3 represents inositol triphosphate, and DAG represents diacyl glycerol. So what will IP3 do? IP3 will increase the permeability, will increase the intercellular concentration of calcium by going into the mitochondria and depleting the mitochondria. If you ask any student what's the mitochondria, they will say it's the powerhouse of the cell. It has multiple effects. It has it's the powerhouse of the cell. In addition, it is also the regulator of apoptotic programmed cell death. It is also a storage site of calcium. And the endoplasmic reticulum is also a storage site of calcium. So it will deplete them from calcium, and then calcium inside the cell concentration, with the concentration of calcium inside the cell will increase, but this time it's not coming from outside, it's from inside. And that will lead the increased intercellular calcium concentration that will ultimately lead to target effects. These target effects could be smooth muscle contraction and so on. Diacylglycerol, what it will do when it's activated is that it will activate for us a very important protein kinase known as protein kinase C. That makes a lot of sense because we're dealing with phospholipase C. So now we'll have protein kinase C, and protein kinase C will go and phosphorylate for us some target enzymes, and these target enzymes will manifest for us our target response. These are all the second messenger systems. So if you have a peptide hormone, this peptide hormone will never ever manifest its target response except through one of these systems that are now known. Either the enzyme linked receptor system, for example of that is the Janus kinase, we talked about that, or the calcium calmodulin system, or the G protein coupled receptor that is divided into GS, GI. There's also GQ, and then there is the phospholipase. Thank you very much for your attention, and I hope that this lecture was beneficial. If you have any questions, please leave them in the comments and I'll try to answer them. Thank you very much, and have a nice one. Take care. Bye-bye.